Hello friends, so we are studying the gram staining, and in last lecture we studied the procedure behind the gram staining. Now let us see what is the mechanism behind the gram staining, how these organisms are differentiated as a gram positive and gram negative. As you know, those cells which are observed as a violet are called as gram positive, and those which are pink are called as gram negatives. Now how this differentiation is there, that was explained by different mechanism behind the gram staining. Now which are these mechanisms? There are four different mechanisms. One is lipid content theory. First and well explained mechanism is lipid content theory. Then next is magnesium ribonucleic theory. Magnesium ribonucleic theory. Stern and Stern theory is third one. Stern and Stern theory. And third is peptidoglycan theory. Peptidoglycan theory. In this particular lecture, we will go in detail about the first two mechanisms of the gram staining, and that is lipid content theory and magnesium ribonucleic theory. So let us see about the study about the peptidoglycan theory. Lipid content theory. Now what happens when you have provided with the culture having both types of the bacteria? Suppose bacterial suspension consists of both gram positive bacteria and gram negative bacteria. Now how this is differentiated during the staining? Here I will write the stain. Now what happens? These gram positive and gram negative bacteria are different in their cell wall composition. In case of gram positive bacteria, their cell wall has less lipid content. So less lipids are present, unsaturated lipids are present in the gram positive cell wall, and to that of the gram negative, percentage of lipid is higher. So higher lipid percentage. That is the basic difference between the two cell wall, gram positive, gram negative. On this difference, on this basis, this theory explains the gram staining. When you apply first stain of the procedure, that is crystal violet, after first stain, your entire cell becomes violet, because crystal violet enters into the cell. It gives violet color to the both cell. Crystal violet is a basic stain, it gives color to both the cell. So after first stain, you get violet cells. Both the cells are violet. Then next is second step is you are applying the gram sardine. Gram sardine acts as a mordant. So it forms a complex with crystal violet and it fixes crystal violet in the stain. And after second step, again both the cell becomes violet. So at the end of second step, again both cells take the color of crystal violet and your stain get fixes with the help of gram sardine, and both the cell will observe as a violet cell. Then in the third step, you are applying ethyl alcohol. As you know, alcohol is good solvent. Here it acts as a decolorizing agent. It acts as a decolorizing agent. It decolorizes, specifically decolorizes gram negative cell. Now how this happens? As you know, these gram negative cells has higher lipid in their cell wall. So all that lipid dissolve in ethyl alcohol. Ethyl alcohol is a good solvent. It dissolves that lipid and there is formation of large pores on the gram negative cell. Through these pores, your CVI complex comes out. So that CVI complex gets extracted from the cell because of ethyl alcohol. Large pores will form higher lipids from the cell wall get dissolved to form the large pores through which CVI complex comes out and your cell get decolorized. Cell become colorless, especially gram negative cell. What happens in gram positive? As lipid content is very less, pores will form, that lipid will get dissolved, but that these pores do not allow that CVI complex to come out from the cell. So CVI complex once stain the cell, gram positive cell, it retain in the cell. So after the third step, your gram positive cell remain violet and gram negative cell get decolorized, selectively decolorized and become colorless. And in last step, you are applying safranin. It is a counter stain. It is also basic stain. Here gram positive cell are already violet. They remain violet. They do not take the safranin because safranin is less basic. Crystal violet is more basic. So it once stain the cell, it remains violet. But in case of gram-negative cell, as these cells are colorless, become colorless in third step, they take the color of safranin in the last step. So that is how after the staining you get violet and pink cell. Those cells which are violet are called as gram-positive. Those cells which are pink cell are called as gram-negative. So that is the simple mechanism behind the lipid content theory. Now next theory is called as magnesium ribonucleic theory. Now it is magnesium ribonucleic theory. This is also another second simple mechanism which explains the gram staining. According to this theory, magnesium ribonucleic is present only in case of gram-positive bacteria. It is present only in case of gram-positive bacteria. It is totally absent in gram-negative bacteria. So what happens when you are applying stain? Again, I will draw the table. Here, when you apply the first stain, second stain is iodine, third is 95% ethyl alcohol, 95% ethyl alcohol, and last is safranin. So when you apply, this is gram-positive bacteria. This one is gram-negative bacteria. When you apply crystal violet, both the cells become violet by taking color of crystal violet. Then you are applying CVI, iodine, so it forms, it acts as a mortar, both the cell becomes violet. Now what happens as this gram-positive bacteria, as you said that gram-positive bacteria consist of magnesium ribonucleic in their cytoplasm. So this magnesium ribonucleic forms a complex with the CVI. There is formation of CVI-MgR complex. CVI-MgR complex, which is more complex than that of the only CVI complex. So there is formation of a large complex due to the presence of magnesium ribonucleic in gram-positive bacteria. Then when you apply crystal alcohol, this CVI complex can easily extracted. It can easily dissolve and your gram-negative bacteria become colorless. At the same time, in case of gram-positive, this large complex, CVI-MgR complex, CVI magnesium ribonucleic complex, is very large. It cannot be easily dissolved by alcohol, and that remains, so your cell become violet. And after safranin, this colorless cell take the color of safranin and they become pink. So that is the mechanism called as magnesium ribonucleic theory. So that is about the first two mechanisms of the gram staining. So in next lecture we will see the second two mechanisms of the gram staining. Till then, stay safe, stay tuned.